published 3 books and 1 book chapter. He has received Best International Researcher Presidential Award 2011 from the Korea Research Institute of Bioscience and Biotechnology, Byeonjan, South Korea, and Professor V.V. Shivarajan Gold Medal in 2020 from Indian Association for Angiosperms Taxonomy for significant contribution in the field of plant taxonomy. He has life member in various scientific societies, reviewer in many international journals, and editor in the Journal of Biology, Vietnam. With this introduction, I would like to invite Dr. Ritesh Kumar Chaudhary sir and take over the session to deliver the talk on the topic DNA-based taxonomy in India, potentials and progress. Sir, please.
Thank you so much, ma'am, for your kind words. And
thank you, sir.
And thanks for organizing this session. Thanks to Dr. Arvind Kumar as well, who has given me this opportunity to speak in front of you people and to interact with the students. So, and yeah, I was there in yesterday's session also, and I was listening to Dr. Prabhu Kumar. It was a wonderful session, but indeed it was dealing with the classical part of taxonomy. That is the most important, the basic part of taxonomy. Today, I'll be highlighting the molecular aspects of plant taxonomy. So to start with, I'll start by sharing my screen first, and then just please confirm whether you're able to see this properly. I hope you are able to see this properly. Could you please confirm it?
Yes, sir.
Yes, sir.
Yeah. So today I'll be talking about DNA-based taxonomy in India. Not particularly in India, but just covering all the aspects, how it had started, the taxonomy, and a little bit how it is going on in India, what we are doing here, and what are the potentials, how we can upgrade ourselves, and what kind of progresses we are making day by day. So before I start, I will just give a little brief about my research institute where I work. I work in the Biodiversity and Paleobiology group of Agharkar Research Institute, which is a DST institute under Ministry of Science and Technology, Department of Science and Technology, Government of India. And here we are working under 6 different streams, under 6 different groups. First group is Biodiversity and Paleobiology, which work on angiosperms, diatoms, fungi, lichens. Then we have a separate bioprospecting unit. We have a bioenergy group. We have a nanobiosciences group. We have a developmental biology group where they are working on 3 model organisms, zebrafish, Drosophila, and Hydra. And then we have a very big genetics and plant breeding group also, where they are working on grapes, wheat, and soybean. And you can go to our website as well, www.aripune.org. And here in our group, our department, Biodiversity and Paleobiology department, we are working on several aspects like documenting biodiversity. We are trying to understand evolution. Also, we are exploring biodiversity for human welfare. Also, we are working toward conservation of biodiversity. And also, we are trying to communicate these things, whatever kind of research we are doing, we are communicating through various aspects. So I hope someday you are visiting us, and whenever you are coming to Pune, do visit us. We have a internationally recognized herbarium as well, with acronym AHMA, with almost 30,000 accessions. So this was just brief about my institute, Agharkar Research Institute. Those who don't know, the Dr. S.P. Agharkar, the institute is named after Dr. S.P. Agharkar, who was a very famous botanist, and he had finished his PhD under famous Dr. Engler, and then during this from UK, and this institute was named after him. So to start with, the flow of my talk would be like, first I'll be talking about, I've divided into 4 parts. First, I'll be talking about phytodiversity documentation, how it has moved from morphology to molecules. And also I'll be talking about how the cladistics, how it has arrived, the advent of cladistics. Then what are the potentials and progress? Then the challenges and the way forward for the students who are studying taxonomy. So let's start with diversity documentation. We know that we have been discovering species for quite a long time. You remember from 1750, almost in the history of more than 250 years. Now the speed by which we are discovering new species has increased a lot. So we have been we are describing more species now compared to the last I think just at a greater rate nowadays we are describing the new species. And every year the speed of new species discovery is so fast now. Every year we're describing almost 18,000 new species. You can go through this website of International Institute for Species Exploration, where they have provided this information. And more than 25% of all known amphibian discovery were made only in the last 10 years. And even not in this, but in the last 2 decades, the number of known mammals has also increased to more than 10%. And you know, many of the discoveries indeed, they resulted from the new techniques because of the arrival of the new techniques in molecular biology. So how it has evolved, you can go through the paper by Davis in 1963 paper. So we started plant taxonomy with describing new species, new plants, naming them, identifying them, and classifying them. And that phase was called exploratory or pioneer phase. After that, we moved ahead and we started making floras, doing monographs, and proposing different classification system. That phase was called consolidation phase or systematic phase. We moved further and we started taking data from from different chemical compounds, numerical, and the numerical taxonomy was introduced. Then we have taken data from cytology, anatomy, embryology, palynology, combining them and proposing a new system of classification and trying to classify them. And that phase was called experimental or biosystematic phase. I think this all you might be aware of. So, but still, I'm just repeating. And presently, we are dealing with the holotaxonomic phase, or this where we are following a holo approach. Like we are taking data from everything, not only the anatomy, cytology, but also we are considering the molecules, DNA, RNA, protein from everywhere, taking the data and trying to classify the organism. So the first 2 phases, they all together, if you see, we call that as alpha taxonomy, and the second one we call as omega taxonomy. And please stop me if you have any question at any time, just raise your hand. I'll be happy to answer. But in the last 2 decades, you might have seen that one of the most exciting developments has taken place. That is the arrival of this molecular systematics. And here, the term we use to mean macromolecular systematics means in plant taxonomy, we are using DNA and RNA to infer relationship among plants. Before going ahead, I would like to ask, usually I ask in offline sessions that how many of you are studying taxonomy? I believe most of you sitting or present here in this webinar, most of you are studying taxonomy, right? So, but how many of you would like to include molecular data to your studies? I hope most of you would like to include molecular data to your studies, because to get higher resolution of your study. Then how many of you believe that your present study will contribute to the knowledge of biodiversity? Whatever you're carrying out presently, suppose you're working on morphological aspect, or you're doing taxonomy of a particular group, you're revising an Ranunculus, or you're revising a particular genus. So how many of you believe that your present study will contribute to the knowledge on biodiversity? So of course you will say, yes, my present study is going to contribute a lot to the knowledge of biodiversity. But please, before going ahead, just you go through a paper by Hotel et al. in 2015, which says there are 7 shortfalls that beset large-scale knowledge of biodiversity. Means there are several shortfalls called Linnean shortfall, Wallacean shortfall, Prestonian shortfall, Darwinian, Roncarian, Hutchinsonian, and Aldolian shortfalls. So you should try and make efforts to overcome these shortfalls. What is Linnean shortfall? So unless you describe each and every species on this earth, there is a shortfall called Linnean shortfall. Unless you understand about the geographic distribution of the species, there is a shortfall called Wallacean shortfall. unless you know about the populations of the your plant group, there is a shortfall called Prestonian shortfall. Unless you know about the evolution of those plants, there is a shortfall called Darwinian shortfall. Then, unless you know about the functional traits and ecological functions, there is a shortfall called Roncarian shortfall. Unless you know about the abiotic tolerances, there is a shortfall called Hutchinsonian shortfall. Unless you know about the ecological interactions, there is a shortfall called Eltonian shortfall. So here molecular systematics help you. If you are including molecular data to your study, I'm sure you'll be able to overcome some of the shortfalls, though they all are interrelated, but still directly they will help you to overcome these shortfalls to your studies. So how to do this? Most of you, I believe, are students at UG, PG level, and some of you must be researchers. So what we do here, we are taking data from plant genomes. I'll be restricting myself to plant taxonomy only, not going to animal and all those taxonomy. So in plants, you know, genomes are there in either nucleus, the DNA is there either in nucleus, in chloroplast, or in mitochondria, right? So we are trying to sequence them, and we are trying to understand how they are varying from each other. So there are certain genetic markers you must have studied in your biotechnology class, that there are certain genetic markers which actually tells you the identity of the plants. They have been divided into 3 groups. One is morphological markers, and then biochemical markers are there, then you have molecular markers. I'll not be going in more details of these, but just to give you a flavor, I have included this slide. So in molecular markers, you have hybridization-based markers and PCR-based markers. Nowadays, both kind of markers are very much popular in several studies, in plant taxonomic studies as well. You have hybridization-based marker, RFLP, you have RAPD, AFLP, SSR, ISR, and so many nowadays. So but I'll be talking a little bit about the site-specific marker. The most popular markers nowadays are there are certain conserved sequences in nucleus which are called the nuclear markers. So there is something called ITS, internal transcribed spacer, which is one of the most successful markers in plant systems. So this, as we know, this nucleus is very big in size, and this is inherited biparentally. But to understand, this is one of the potential tools to understand phylogeny of a particular plant group. So what we do here, we target a very small region called, suppose there are so many regions, conserved site, which can work as a marker. But as you see, ITS, it can tell you, it has been used widely and still in use to understand phylogeny as a barcode. So this region, if you simply sequence, they're just simply 600, 800 base pairs long regions, but they are so much conserved to that species that they're going to tell you the identity of your plant. Similarly, in chloroplast genome also, you have several specific conserved regions which will tell you the identity of the plant. So here we are not going to sequence the whole chloroplast region, rather some small regions we are going to sequence and trying to correlate with or other species. If some doubtful species are there, we are amplifying these regions, we are sequencing these regions. We'll suppose RBCL, small region, we are just sequencing them and comparing with other species. And if your one species in doubt is the species in question is different, then it will show some sort of variation. Similarly, in mitochondria also, you have certain region, which is, again, there are certain regions like ATP1, ATPA, something like that. And then you can simply sequence them, and after that, you can sequence them and try to understand the identity, phylogeny, and so many things. So just going to the basics. Sorry. So in DNA, what you do, you are going to simply isolate DNA from the plants first, And then after that, once you isolate the DNA, you are going to amplify a particular target region. So because sequencing of the whole DNA is nowadays is very difficult, it's not so easy. So what we do for our convenience, we simply amplify a target region. As for example, the ITS or RBCL, what I said, we simply amplify a region using PCR, then sequence them and get the sequences curated, align them, and after that we construct a phylogenetic tree using some softwares. There are so many freely available softwares online. So after that, you can construct your phylogenetic tree like this, and you can try to understand the phylogenetic position of your particular plant group or a specific plant. So to make it more clear, you take whole DNA from here, and then here is the primer for that particular region. Like for ITS, you just add this primer and you put in a PCR premix, put in this PCR, amplify this and send this to sequencing agency. Sequencing agency is going to send you the sequences using their sequencer, and you have to use this data to construct your phylogenetic tree. This is a very old one, PowerPoint have used, but now there's so many different softwares have come using which you can construct your phylogenetic tree. Then how actually it has arrived, the science of cladistics. So it is very interesting. I'm sure you have seen this this illustration, and I don't know how many of you have seen, but this is a very interesting phylogenetic tree, I would say, which was made by none other than the great Charles Darwin. And this is when he visited the Galapagos Island, he saw that in different islets there are several birds, the Darwin's finches, they have variation in their beak. Some of them were having a very sharp beak, and some of them had a very blunt beak. So based on their morphological characters, he constructed a phylogenetic tree, and he wrote, I think. So that this particular thing, the same concept has become so famous that it is known as the famous I think tree depiction by Charles Darwin. From here, the concept of cladistic actually arose. But it was still not very popular unless Willy Hennig in 1950 he published a book. He was a German entomologist. He published a book in German, which was later in 1966 published as a phylogenetic systematics as a book form. And there he proposed the concept of cladistics. Cladistics has come, the word has come from the Greek word called klados, means branch. In here, he has proposed that it is an approach to biological classification in which organisms are grouped together based on whether or not they have one or more shared unique characteristic. So I think these things you'll be understanding later, what is the meaning of shared unique characteristic that come from the group's last common ancestor and are not present in more distant ancestor. They are talking about the character evolution and how they're being shared in a particular group. So he proposed this idea of cladistics in 1950 itself. And here, what is special that here you are not only using to construct this relationship, you are not only using the morphological data, rather you can take data from either the paleological data, palynological data, morphological data, anatomical data, embryological DNA data, everything you can take, put together, and you can construct your cladogram. And then you can try to understand the relationship of the different species. And here, when we talk about characters, it's not only about the morphological characters. Here you can combine physical characters along with nucleotide sequences in the behavioral traits as well. How in different ecological situation, the plant behaves, and combine all the data together and try to understand the relationship. Same way, and the characters, when you try to understand, the character means they are either in binary form or in multi-state form. Binary form means the characters are whether present or absent, and multi-state means whether they are the same character is present in 3 different forms. If I'm talking about binary characters, it means either in the form of 0 and 1. So as you can see, the horns, they are absent in horse, and the antlers, the presence in horse, but they are present in deer, but they are absent in horse. So this is a 0-1 character, whether the same character is absent or present. But some of the times, in many of the times, you will see The same character is present in different forms. Suppose I'll give you the example of hooves. The presence of hooves in horses, they're one, whether in caw it is divided, it is two in caws, and in human being there are five. So same character, but they're present in multiple different forms. That's why such characters are called as multi-state characters. Here we call binary characters, and if the same character is present in three different forms, they're called multi-state characters. And then using those characters, combining all those characters, we prepare a matrix. Suppose you're dealing with 4 different species, one of them having eyes but not spines, some of them having antennae. But so you make a matrix, and using that matrix, you can construct your phylogenetic tree. So since I'm not able to communicate properly with you, but still I will just try to explain this. In the following cladogram, you see, the question 1 is in the following cladogram, which node occurred earliest in time? So this one we call node in a phylogenetic tree. This is node 1, node 2, node 3. So the first question is, in the following cladogram, which node occurred earliest in time? So you can see this node actually appeared earliest in time. So your answer would be, okay, node 1 appeared earliest in time. Then you see which node represents the most recent common ancestor of terminal taxa B and C. Terminal taxa B and C. Which one would be the ancestor? 1, 2, or 3? You can see 2 has given rise to B and C. That's why your answer would be 2. I'm trying to explain you how to read a phylogenetic tree, nothing more than that. Then the third question is, which node represents the most recent common ancestor of terminal taxa A and B? Which node represents the most recent common ancestor? Most recent common ancestor of terminal taxa A and B. So this node, we are talking about node 1. This is the most recent common ancestor of terminal taxa 1 and terminal taxa B. So similarly, if you see our plants, how they have evolved. So you see the same way. If you see the angiosperms, gymnosperm, bryophytes, algae, they all have the evolution in their characters. So these characters we can consider as primitive, and as you move to the higher plant group, the characters is getting advanced, and such characters are called derived characters. So this would be the most primitive group, and this we consider as the most advanced group. So to explain it further, you can see nodes actually represent the end of the ancestral taxon. It means the nodes is the end of the ancestral taxon, and the stems, the species that splits from the ancestor. So for human and gorilla, node C is going to be the ancestor. 2 taxa that split from the node are called sister taxa. So 2 taxa, suppose from this node there are 2 species or 2 particular group have arisen, human and gorilla. So you can call them as a sister taxa or sister groups. So now focus on node C. At the node, the ancestor goes extinct but leaves 2 siblings hypothesized to be humans and gorillas. Humans and gorillas are sister taxa and more closely related to one another than either is to chimpanzees or baboon. So if I ask you, gorilla is most closely related to baboons or to humans, so your answer should be humans. Same way, if I ask you, angiosperms are related most closely to gymnosperm or to algae, so your answer should be, it should be gymnosperms. At node A, we find the splitting event, right? At node A, you can see there is a splitting event that led to the baboons. And then you have chimpanzees, humans, and gorillas. This way, we are able to find out that chimpanzees, human, and gorillas are more closely related to each other than to the baboons, right? In this example, this particular group you can take as an out group. You can consider it as an out group, which is distantly related group. And this would be called as in group. Now we'll be talking about the potentials and progress. What kind of potentials are there and how much progress we have made. So you might have heard about the numerical taxonomy also, which was used almost, I think, 20 years before. There were several papers, several research they were carried out to understand the relationship using the morphological characters in a quantitative form. So suppose we used to make a table. If the character is present, just check the morphology and you see if it is present, you can simply put 1. If they are not, then you make 0. And then you try to understand their relationship, like A is most closely related to C based on the numerical data you get. Suppose the particular gland is present or absent, the leaves is linear or ovate, that way you make the matrix like this, and after that you try to understand the characters. But this particular study, which we call as morphometry, phonetics, or numerical taxonomy, that has a problem. I'll just try to elaborate this with this illustration. So suppose Opuntia is a cactus, and then you have Euphorbia. This belongs to Euphorbiaceae family. Same way, Pereschia is actually Cactaceae and Codium is Euphorbiaceae. But if you compare the morphology of them, then based on morphology, you can say these two are more closely related to each other. Opuntia could not be closely related to Pereschia because their morphology is quite distinct, quite different. So the phenogram will present the relationship like this. But if you take out DNA from all of them, you can see that Opuntia and Pereschia, it will show you as a most closely related group than Euphorbia or Codium. So that's why we rely more on the molecular data. And this numerical taxonomy was nowadays not much popular. Then the example which I showed you, sometimes, one of them belongs to Cactaceae, another one is a Euphorbiaceae. But because of some ecological pressure, this is showing the characters of a cactus. So in such cases, if you're doing numerical taxonomy, you might end up messing up the things. So that's why if you are taking DNA out of them, it will clearly tell you that this actually belongs to Euphorbiaceae, not to Cactaceae. Then this DNA-based data has been helping us to classify the organisms, to classify them in particular groups. You might have heard about APG1, which was proposed in 1998, Angiosperm phylogeny grouping, and where it was proposed that there are 40 orders and there are 462 families. And we advanced further, and then APG2 was proposed in 2003. After that, APG3 also came. As we have more and more data, more and more DNA data from the plant groups, we are kind of revising the taxonomy, revising the phylogeny of the particular groups. And now, presently, we have a PG4, which tells us that there are 416 families under 46 orders. So you can see how molecular data is changing the taxonomy, changing our concept, changing the phylogenetic relationship, in our understanding of the phylogenetic relationships. Then the DNA data also helps us in barcoding. So why this barcoding is done and why it is so important? So, you might have seen that when you go to shopping mall, you see that there are several specific barcodes on some products. It means it will tell you everything about that particular product. Same way, if you are developing a barcode for the plants, so you're not going to develop something like this, but you are going to isolate those particular sequences. You are going to find out those particular sequences, which will tell you everything about the identity of that particular plant group or the plant. So it helps us in true identification of or for proper classification. So it will help you whether whether suppose you are studying some Lamiaceae groups or particular genus Ocimum, suppose. So whether it's real Ocimum or something else, if you're doing barcoding, if you're using those barcodes, it will tell you with surety that this particular species belongs to, this particular DNA sequence belongs to Ocimum sanctum and not anything else. And also it helps you to stop adulteration in herbal medicines. I'm going to explain this later also. So, there are so many similar looking species. So in herbal medicines, what happens, a simple layman goes for collection and simply he thinks that, okay, this could be the same species. He collects different species and mix them in your herbal drugs. And as a result, your medicine is not effective. So if you are using DNA barcode for herbal medicine, for authentication of herbal medicine, that is going to be very, very useful. Also, it helps you in conservation. So suppose sometimes what happens that we we are conserving a wrong species instead of what we should conserve, because we don't know the true identity of the species. There have been several cases where a wrong species was selected to conserve, and people have spent a lot of money to conserve a wrong species instead of what has to be conserved had to be conserved. So this, if you're checking that with DNA barcode, what actually we should conserve, this will help you a lot to find the proper identity of the species. So what we do here, we are using again the same conserved nucleotide sequences to identify a taxon. And this can be done using two approaches. One we call Loci-based barcoding, where you are taking data from nucleus or mitochondria or from the chloroplast, and you are comparing those data, and then you have to establish a standard barcodes to standard barcode. There are several statistical analysis using which you can establish a DNA barcode for a particular species. And there is another approach called plastome-based barcoding, where you have to you have to sequence the whole chloroplast genome, and based on that you can propose some specific barcode for a particular species. I will tell you a very interesting case of a particular plant group, Eriocolon, on which we have been working for almost more than 5 and 6 years, last 5 and 6 years. So this particular group is called Eriocolon. I think in Tamil they are called as Mikpu, because the head looks like a microphone. But if you're just going to check them on a herbarium sheet, they're so difficult to identify. These three, actually, they belong to 3 different species. But in a herbarium sheet, they all look alike. So unless you're going to check the heads, the character of these heads, you cannot confirm the identity. And there in India, we have almost more than 100 species of Eriocolon. And many of them have proved effective as neuroprotectant. And they have also demonstrated significant therapeutic effects on headache, toothache, nasosinusitis, and eye diseases. So, but unless you confirm the true identity of the particular species, how you're going to use it, how you're going to understand which species is going to be a good neuroprotectant. So, but identifying them is also again very difficult. What you have to do, once you want to identify them, you have to dissect the flowers, the heads, and you have to check the characters of involucral bracts. You have to check the characters of floral bracts, then male flowers, then female flowers. And this is not enough. You have to check for the seeds as well, which are very minute. Light microscopy will also not help you, because unless you see them under scanning electron microscope, and that also not as a whole, but unless you see the presence or absence or the structure of the appendages which are present on the seeds, you cannot confirm identity of most of the Eriocolon species. So we thought this would be very challenging group. We conducted molecular systematic studies on this particular group, and we came up with very good data set. And and also among species also, they show a lot of variability. So in the same species, you can see a lot of variation. That's why this has been referred as taxonomist nightmare. This plant group is so difficult. And even if you see Flora of British India, the great J.D. Hooker has written that they are the most difficult of classification, presenting no good sectional characters. In India, the work on Eriocolon was carried out by Ansari and Balakrishnan-ji, but that work was totally based on morphology. So we wanted to check the classification system, identity of the species based on the molecular data. And for this, we collected a lot of accessions from all, like most of the Western Ghat, the area, and also in the Northeast region. And then we collected multiple accessions of them. And then we came up with the phylogeny of this. This is just a representative diagram, not the true one. Recently, we have published the phylogeny of this particular plant group in a very good journal, Biodiversity and Systematics and Biodiversity. And also, we proposed DNA barcode, which will help us to correctly identify these species. And this work was also published in PLOS ONE. We are working on another plant group called Capparis. You might have seen this. There are several species in Tamil Nadu as well, and also in Kerala. So this particular Capparis, in Ayurveda, they are referred as Himsra. And this is, this is one of the major ingredient of the famous medicine Liv52. And in India, we have almost 31 species of this group. And we thought, okay, we should propose DNA barcode for all these species because many of the species, they look alike. So suppose if we want to collect Himsra, but how to identify them? Because many of them, they look similar. So we have made effort to propose DNA barcodes for them, and this will definitely help us to check adulteration in herbal medicines. In same way, we have proposed Loci-based barcoding and also plastome-based barcoding. And they are also very famous. The fruits are eaten. They are very famous as a pickle in European countries. So this way you have seen the importance of the DNA data. So this is not enough. The DNA data also helps you to understand the ancestral state, understand the ancestral state, which means how, suppose you're studying a particular genus, many species are there, you can see how the characters were in their ancestors. So using DNA data, you can try to understand how a particular specific character has evolved in this particular group. As you see, suppose I would say in Lamiaceae, you can see the verticillus inflorescence, right? That is very unique to Lamiaceae, not to other groups. So how that particular character has evolved especially in Lamiaceae, if you're taking samples, DNA samples from different plant group, you can understand how this particular character has evolved with surety. Not only this helps you to understand in different how they have evolved in different geographical regions and how many years before. Suppose if you're taking DNA data set and combining with this fossil data, this will help you to understand where the species originated, in which continent, where it originated, and how it spread throughout its present range. And there are several free softwares available using which you can try to understand. And you can see two distinct vertical lines here, which actually tells you about the age of that particular group. So you can tell, okay, 30 million years ago, the state of this particular group was like this, and it has kept on changing in different, like in million years ago, this scale, how it has changed. Then it also helps you to understand the cryptic species. What are these? Cryptic species, they are morphologically alike species. Like if suppose you're working on Ranunculus diffusus. So some of the species of Ranunculus or some particular group you will find in nature, in nature, if you go for collection, they actually look similar. Morphologically, you will not find any variation. As we have faced this in our areocolon study also, some of the areocolons, they look alike in feel, but you just isolate DNA from them, you check the sequences, they are different. The molecular data says they are different, but morphology, they are not at all different. So those kind of species, they are called cryptic species. So DNA data also helps us to understand the cryptic species diversity. And there are several papers available online. You can check and you can read and try to understand this. Then nowadays we have next generation sequencing also, where you can use whole genome data or whole plastome data for phylogeny and barcoding, as I already discussed before. So what happened, how it helps next generation sequencing? Earlier it was very difficult to sequence the whole genome because of the cost involved, because of the time and effort involved. But nowadays we have very good sequencers available in the market, and even the university, they have big, like you have Illumina platform, HiSec, MiSec, everything is available. So you can have better resolved phylogeny, and it also helps us to deal with the difficulties posed by polyploidy and hybridization. Because of these issues, we sometimes wrongly identify the species, and also to understand the species boundaries and their relationships. So this is one of the plastome sequence which helped people to understand the phylogenetic relationship. There is a very famous lady taxonomist called June Wen. If you see how the things has evolved, in 2001, she publishes a paper in a very good journal, Systematic Botany, 2001, but the paper was simply based on ITS data set. Simply one particular nuclear region, and you can publish your paper in systematic botany. But it was possible at that time. If you move further in 2004, you can see the same lady, she has published using data set from ITS as well as chloroplast. So you are improving, upgrading yourself, your work, and this work again was published in Plant Systematics and Evolution Journal. Then in 2013, the same group, they have published their data in PLOS ONE using chloroplast genome. So starting from ITS to chloroplast genome, how the journey in 10 to 12 years, you can see it has evolved. And they have, using these data sets, they could resolve so many complexes. There were several complexes in Araliaceae family which they could resolve using plastome data sets. But things are just evolving each and every day. Nowadays, people are using more and more, like 250 plastome data or more and more plastome data, putting them together and trying to understand the relationships. Then population genetics has always been a problem. Suppose you want to understand that between population and within population, what are the variations? There are nowadays several techniques called RADseq, GBS, genotyping by sequencing methods, restricted site associated DNA sequencing methods, using which you can try to understand the population genetics of a particular plant group. And they help us a lot, and they are also low cost. You don't have to go for the whole genome sequencing. They're also low cost, and they can help you in understanding the ecological and evolutionary genetics or something, whatever you are interested in. So this method has again been proved useful for so many cases. Here, what you do, you use restriction and endonuclease, cut the whole genome in several regions, get SNPs, and they will help you to understand the population genetics, understand breeding. They're helpful in breeding and also trait mapping diverse organisms. There is a very famous paper, very good paper on coffee is available. You can go through this paper. I just wanted to cite the example of this paper, which has helped us a lot. Everyone knows here coffee, but can anyone answer here how many species of coffee are there in the world? Can you simply make a wild guess? 2, 3, 4, 5? No. There are 124 species of coffee in this world. But the crop coffee, the one which is being used as our regular coffee, our coffee comes from two famous species called Coffea arabica and Coffea canephora, where you have high caffeine content. And one of the most allied group is Silanthus, where you don't find caffeine at all. So these fellows, they have tried to understand the relationship of all those species, how the caffeine has evolved. That also they have tried to understand in this Coffea group. And using the same GBS technique, using the same SNPs, they have constructed a phylogenetic tree where they found there are two distinct clade called xenoclade and euclade, where you can see, interestingly, this particular almost 50% of the group of the coffee species, you won't find caffeine at all. But the caffeine is present in rest of the 50% species, mostly belonging to Madagascar. So they came up with this interesting observation, and also they told that how this particular coffee genus has evolved in the world, in which region, which continent actually it started evolving, and how the characters started changing. And then how the caffeine content has evolved in due course of time. They have also, you can see this red line, it means the highest caffeine content, and blue line means there is no caffeine. So you can make out that how this caffeine content has evolved, right? And then there is a very good paper available in systematic biology, where they have used the same kind of the restriction and site using the restriction endonucleases, and the same method. There was a very good paper. There is a very good paper available on cryptic viburnum species. Viburnum, already you might have seen it, but it is also found in North America, and they are discussing with 3 different viburnum species, where the three different species, they're so related to each other that there are a lot of conflict among taxonomists, whether the two belong to the same species or they are three different distinct species. And they are simply different by each other by their margins. One of them having a dentate margin, another is simply, you can see entire margin and little undulate margin. So there were there are a lot of conflict whether they belong to the same or different. But this particular technique, they help us, this RAD sequencing, they help them to understand how these three are different from each other, and they came up with a clear-cut classification. Also, we have nowadays something called genome skimming technique available, where you don't have to sequence the whole genome of a plant. Rather, you can simply skim. You can use the skim data. You can cut them in different fractions, and using those fractions, almost 5% of that particular region, you can generate fragments, and checking those variation in that particular 5%, you can come up with your classification system. You can tell whether they are same or different. Then also this concept is being changed. Nowadays, you have something called geogenomics, where you are going to integrate the data from geology, geography, genomics to study the coupled evolution of life on Earth. This is again an emerging field. It employs genomic data to solve geological problem or constrain geological hypothesis, where what you are going to do, you are going to, and using these you're combining these data, you can try to understand, or you can provide chronology for past geological events. Using this sequence data, DNA sequence data, see how interesting it would be. So using your taxonomic knowledge, using this DNA data, you are going to pose chronology for past geological events which has taken place. Also, you can talk about, you can interpret the paleo-environmental interpretations. You can interpret the data in that way. But still, there are several challenges, particularly in a country like India, but there are still way forward. I'm just going to discuss that. People, if you're studying plant taxonomy, they will consider you, if you're studying plant taxonomy, it's simply a basic science. You're simply going to understand morphology. There's no science involved. There's so many people. But if you see that in 1968, this thing was proposed in Nature, there's a paper published called "Is Botany Dead?" But you see, presently, till date, taxonomy and botany, they're growing so well, they're doing so good. Almost 50 years ahead, we are doing great in plant taxonomy. So don't worry about this, whether if someone tells you plant taxonomy is going to be dead, it's not a good, like it's an old-fashioned science. So don't do that. Don't believe them. There are several papers, and just try to read this. I would like to quote this sentence from Sidney Bassett from UK. He has said, as long as there are people on Earth to feed and an environment fit for plants and humans to grow in, the service of botanists will continue to be needed. If today the function of botanists in society is in question, then university departments must carry some of the blame. For without doubt, many potential and existing botanists, not to mention the general public, are struggling through the trees looking for the wood. So yes, I should not directly, but this actually tells what are the responsibilities of a taxonomy teacher, and if we are teaching that to our students, definitely we are going to excel in our field. You can also show this to people that see all these work which we have discussed here. They have published in a very good journal. Like this viburnum work was published in a journal having impact factor 10.4. The Coffea was published in impact factor 4. Same thing, this has not discussed, but still, if you want to publish a new species, if you take data set from different technology, of course you can publish in a high impact factor journal. In India, you can go through this paper which we had recently published. We have reviewed the status of molecular systematics of flowering plants in India, and you can see we have a rich diversity of plants and also a high degree of endemism in the country. But the lineage or evolutionary lineage has not been properly understood. And we have been working, and there are several groups who have actually worked on Cucurbitaceae. We have worked on Eriocolaceae. There are several groups who have worked on Leguminosae, Piperaceae. They have been worked on to some extent. They have been worked out well, but still there are several groups like Orchidaceae, very important group, Poaceae, Rubiaceae. They have not been worked out till date. So it's now the duty of yours to look into the phylogeny, take more, collect more and more DNA samples, DNA data from them, and try to understand the relationship of those, particularly which are native to our country, endemic to our country. Because if, because because of biodiversity laws, outsiders, they cannot take DNA, cannot use this DNA data set. It is only you who can use this DNA data set, and you can propose a new classification system. You can help them to understand their phylogeny. So what are the challenges in Indian scenario? In universities nowadays, we are giving too much emphasis on the detailed life history of selected represented plants and less on the issues of environmental relevance. So how they're actually these lifestyles, suppose you are reading the life history of a particular plant, how they'll be relevant, that also has to be taught. Then there are less exposure to fieldwork. People, they finish the plant taxonomic studies in lab itself. That should not happen. There's a less exposure to advanced tools and techniques. Many of the labs I have seen, they have not even seen the gel electrophoresis unit. They have not seen the PCR. So we should get more and more exposure to the advanced tools and techniques. But also we can blame it to funding agencies which provide less funding for taxonomic research. We have very stringent biodiversity laws which also prohibits us to carry out plant taxonomy research. Then also we have certain hindrances in international collaborations. But don't be sad, there's a way forward. Nowadays, SERB has come up with certain things, something called SSR, Social Scientific Responsibilities, where you have to train, if you have a project on plant taxonomy, you have to train to some student on the plant taxonomy project and all. And you will get some funding support as well. And similarly, as it was proposed, PBI was proposed in the USA, Planetary Biodiversity Inventories. Same way in India, the National Biodiversity Mission is being planned, under which a lot of emphasis will be given on taxonomic research. So the roles of taxonomist is being changed. Now it's not only related to determining the identity of a plant, though not only related to establish and revise the taxa, but also you have to build the phylogenetic systems. You have to integrate the information from different fields to build species biogeographies. You can create identification tools, and also it's your duty to train the next generation of taxonomists. So to conclude with, I would just this concluding slide, I would say that even if you are not carrying out molecular taxonomy, don't be disappointed, because for some of the countries like India, some of the developing or tropical countries like India, Vietnam, Cambodia, the alpha stage of taxonomy has not been passed. So a careful, thorough, competent, descriptive taxonomy can also reveal fascinating details of the evolutionary history. So even if you are studying even if you are carrying out plant morphology-based research, don't be disappointed. You can still publish good papers, you can still talk about evolutionary history, but yes, you have to keep on updating yourself. What are the changes going on? What are the new tools and techniques are coming in the field of taxonomy? You have to keep on reading, you have to keep on checking the things. So to conclude with, I would say that molecular taxonomy, actually, it's not a substitute to classical taxonomy. It's not going to change classical taxonomy. Rather, it's complementary to it. It's still a part of taxonomy. What we are doing, we are supporting the classical taxonomy with the tools, with more and more tools. Recent taxonomic papers, they include DNA information, but it is not like that. The morphological data characteristic has not been discussed. You have to discuss both morphology in light of the DNA data or DNA results which you have got. So it's not going to replace the traditional taxonomy. That is going to remain there, but we are going to support classical taxonomy with DNA data sets. So if someone asks you in Dr. R.R. Rao's paper, he has asked whether it's time to be chemo, cyto, histo, eco, morpho, molecular taxonomies. So yes, my answer is yes, yes, sir. This is the time to become a chemo, cyto, histo. You have to combine everything together and then you propose something with a good classification system. So this is a cartoon I have shown here where two frogs are talking, that looks are not everything. It is what inside you that matters. A biology teacher told me about this. With this, I would like to acknowledge this to our director, Agarkar Research Institute, and also to Dr. Aravindan for inviting me, and also to my molecular systematic lab team at ARI. Thank you so much. If you have any question, definitely you can write back to me on my mail ID, which is given here, rkchaudhary@aripune.org. So let me know how did you find this talk, and I'll wait for your feedback. And if you have any question, definitely you're also welcome to just ask it now. Thank you so much.
Thank you, sir. The topic is now open for discussion. Participants, you can ask your questions to the speaker. You can also post your queries in the chat box.
Yes, I can see. Yes, you can post your questions in the chat box. I'll be happy to answer them. Yes, Dr. Arvindan, are you there?
Yes, sir. Yes, sir. Thank you, sir.
Yes, welcome. So I hope you find it informative. I hope the students will like it.
Yes, sir. It's very informative, sir. Really enjoyed the session, sir.
Thank you so much. I hope I'm expecting some questions from the participant. And Dr. Keith, anybody telling excellent. Thank you, doctor, for your comments. And if any student, if they have any questions, I'll be happy to answer. And also you can write me on this, my email ID. Suppose if you are hesitant in asking on a public platform, definitely you can write back to me. If you have any sort of question, any small question also there, you're welcome to ask this. Okay, again, I'm writing my email ID in the chat box, rkchaudhary@aripune.org. So you can post your questions here as well. And if you want a copy of this presentation, I'll be also happy to share this with you. So write me back on this email ID if you want a presentation. Huh? Thank you, Dr. Raja, Dr. Ricker. So I'll stop sharing my screen then, Dr. Aravindan, is it fine?
Yes, sir.
Yeah.
Thank you so much, sir, for your elaborate talk on the topic. Let us
Thank
be
you.
grateful to the people who make us happy. They are the charming gardeners who make our souls blossom. I now welcome and invite Dr. V. Aravindan sir, Assistant Professor in Department of Botany, Konginodo Arts and Science College, Coimbatore, and the Organizing Secretary of this national webinar to express the gratefulness and to make the souls blossom. Please, sir.
Thank you, Betty. Good afternoon to one and all gathered here. I deem it a great honor to propose vote of thanks to all who have helped us in making this webinar such a resounding success. First of all, I would like to propose hearty vote of thanks to our chief guest, Dr. Ritesh Kumar Chaudhary sir, Scientist D and Senior Scientist, Biodiversity and Paleobiology Group, Agarkar Research Institute, Pune, for gracing today's webinar. Thank you, sir, for your very interesting and thought-provoking address, and also to take out time from your busy schedule to grace this event. Today's session was full of knowledge and interesting things. It gave deep insights into the topic and also revealed some interesting facts in molecular taxonomy. I would like to thank our distinguished yesterday speaker, Dr. K.M. Prabhukumar sir, Senior Scientist, Plant Diversity, Systematics and Herbarium Division, CSIR National Botanical Research Institute, Lucknow, for making excellent presentations and making this webinar interesting and meaningful. Thank you, sir. On behalf of Department of Botany, Communal Arts and Science College, and my own behalf, I extend a very hearty vote of thanks to respected Secretary cum Director, Dr. C.A. Vasing Madam, for her support and encouragement. Thank you, Madam. I would also like to thank our Principal, Dr. M. Lachmanasamy sir, for giving permission to organizing this webinar and inviting resource persons to conduct it. Thank you, sir. I also wish to express my gratitude to Dean R&D, Dr. S. Palsamy sir, Dean Academics, Dr. S. R. Madan Sangar sir, and Dean School of Computer Science, Dr. N. Nagarajan sir, for their encouragement and support. Thank you, sir. Also, I would like to extend a special thanks to Dr. V.S. Ramachandran sir, retired professor, Department of Botany, Bharata University, for inspiring us to do this program. I would also like to thank our beloved HOD, Dr. K. Tenmoli Madam, for her moral support and guidance. I must mention my deep sense of appreciation for Dr. Rekha Madam for her constant cooperation and support. As no program can become successful with a single person, so I extend my big thanks to our department faculty members for their support. Thank you, colleagues, for making this event successful with your contribution. I am happy to express vote of thanks to other department faculty members of Congonal Arts and Science College who have made this webinar a grand success. I would like to extend a special thanks to Mrs. Betty, our department research scholar, for her nice and excellent MOC presentation. Thank you, Betty. Finally, the wonderful participants who have turned up in such a great numbers, not only from our department, but also from other colleges and other institutions. Thank you so much for your participation. Once again, I thank all for your cordial cooperation. Thank you.
Thank you so much, Dr. Arvindan, for
Thank you,
inviting
sir.
me. It was a pleasure meeting with you, you all of you.
Yes, sir. Thank you, sir.
Yeah.
Thank you, sir. Few announcements to the participants. Participants are requested to fill up the feedback form provided in the chat box before you leave this session. You will all receive your participation certificates through your registered email IDs within 10 days. For any other details, you can contact our organizing committee. When you practice gratefulness, there is a sense of respect towards others. I thank all the participants who participated in this national webinar and make this program a grand success. Thank you all once again. All reserved and unreserved credits goes to Almighty. Thank you. Have a nice day.